it's not for biology one, then you might need to go to a different room. I don't want you to miss any minute of this because it might be important for you in other class. So just to make sure, this is for biology one, okay? So it might, like many of us didn't have any printout, and maybe, you know, there's some miscommunication or something with, you know, the SARC. So what, if you have a laptop, you can go to UCF studyunion.wordpress.com, and under biology, there will be, you know, the session that different tutors, you know, listed on there. And under in my section, there will be practice exam, which is available on here. But again, if you don't have it, you don't have the computer, you don't have the printout, just go along with what I have. So what my plan today is, I have the 50 questions for the practice exam, and we're going to go over this, and whatever details that I could possibly give you the information about it, I will try to do my best to give you that. And the answer also posted on there, so even if you don't have it, so you can always, you can go home and try to practice on your own and check your answer on there as well. Okay, so let's, it's going to be a couple more minutes before we start. The UCF WordPress, UCF studyunion.wordpress.com. Maybe I'll write it up here real quick. Okay. So that's a website if you have the laptop with you. Okay. All right, so I'm going to start going over this practice exam. Okay, so the first one, before starting, actually, I apologize if you go and find some typos. It was kind of in a rush for me. So there's some typos on there, but we're going to go over it, and I will, I guess, point it out which, or where the typo would be, okay? But the first one, which of the following are not found in a prokaryotic cell, okay? So maybe, you can answer along. So what's the difference between a prokaryotic cell and eukaryotic cell, okay? So anybody know what's the difference? Okay, so if we look, the major things here is prokaryotic cell doesn't have the organelle that has a membrane bound, okay? So no membrane bound organelle. So if we look at here, so we have the, if I just put the difference between prokaryote and eukaryote, okay, is the, let's see, membrane bound. Okay, so if we know this is the major difference, can you identify which one has membrane or which one has not? Then if you can answer this question, okay? So if we look at this, a ribosome, does it have membrane? If you don't know, we can move on and look for the next one, the next one, nucleus. Does nucleus have membrane? Yes. So this one has membrane. Mitochondria? Yes. So if these two has membrane, so we can also look at other answer, right? So these two have membrane. These two has to be in eukaryote, right? So these two found in eukaryote. So these two cannot be found in prokaryote, right? So the answer to this is what? B and C. Make sense? Okay. So that's the first one. Okay. Second one. A covalent bond can be found in which of the following molecule? Okay. So first, if we look, the difference between covalent bond and other bond, the two major intramolecular force is covalent bond, and the next one is ionic bond. Okay, so there's two major types of bond that we're dealing with right now. So covalent bond, what's the key word that we use here for covalent? Sharing, very good. Sharing electron, right? So the other one, ionic, would be transfer electron. So in here, if we look into these three choices, the first one, sodium chloride. Sodium chloride has, sodium is metal, right? So sodium is metal. If we have this metal, and sodium has one valence electron, right? So we have, if we look at this as sodium like that, we have one valence electron, and I'm going to put in a different color. We have chlorine has 7 electron like that. okay? But we know that for metal, the group 1, so we try, in any type of atom, we try to meet the octet rule or duet rule, if you recall back then from at the beginning of the semester. So all of these trying to meet or fulfill the octet or duet rule. So with this only one, we need the sodium tend to lose that electron. So that particular electron right here transfer to the chlorine, so to form the ionic bond. So this one, sodium chloride, is actually has the ionic bond. Okay, so for oxygen, we have, let's see, O2. Oxygen has 6, right? 6 electron, and also it's all O2, so O2, it would form like that, okay? So, or this one, it's a Lewis structure. So we have this, so this one, for this oxygen, we have 1, 2, 3, 4, 5, 6, 7, 8, so that 8. And also this one also had 8. So this octet rule, and these is where they're sharing. So the O2 is has a covalent bond. Okay, so this one has covalent bond. What about water? Water is just similar. Water, we have oxygen and we have hydrogen, right? So oxygen, this 6, and we have hydrogen like that. So for hydrogen, because hydrogen has only 1 electron, so they use duet rule instead of octet rule, right? So the duet rule are fulfilled with this part, so just 2. On this hydrogen is the same. Okay, so with oxygen, 1, 2, 3, 4, 5, 6, 8. So it's 8 is octet rule. So we have this one, since hydrogen and oxygen, they're sharing electrons, so it's covalent. So this, the answer to the H2O also has covalent. So the answer to this is E, just B and C. Okay. So next, which of the following is true regarding hydrogen bond? Okay. So this one, we have the first one, it provides a cohesive between water molecule. Okay. So this one, cohesive, provide the cohesiveness. Cohesiveness, talking about how the molecule of the water, you know, like bind to each other. So this one, if you look at this, you know, this one is how if we look at this, okay, so this, the cohesiveness is between the water molecule. That is, that's correct. So the second one, it provides an adhesiveness between water molecule. Adhesiveness is between water molecule and other substance, not among themselves. So if you recall from reading the chapter, it would be we have the meniscus, or if you go and have some lab before, you see that in the capillary you have the meniscus. So because they adhere to the tube better, so the tube has higher affinity. So the surface of the water come up a little bit just like that. Okay, so I guess this one, the answer, this one it's not correct because this is not between water molecule. So it's between water and other substance. So this one is not correct. Okay. It is an intermolecular force. Is hydrogen intermolecular or intramolecular? Okay. So if we look at this, this is water, right? We talked about before. If this one, because of this lone pair of the water, so make the charge is partially negative. Okay. So for hydrogen here is partially positive. So if we have another OH like that, so this one will also be, can you see it? Let me move it a little bit. Yep. Okay. So this one will also be partially positive, partially negative. So what does it do when you have positive and negative together? So it attracts each other, right? So this negative charge of the oxygen right here is building the bond. This bond is the hydrogen bond, okay? And this hydrogen bond is between molecules of water. So this one is intermolecular force. So this one is also correct. So the answer to this one is D, which is A and C is correct. Okay. Next one. If a solution has hydroxide concentration of 0.00001, then the pH is equal to what? So first of all, when we do the pH problems, so the two major, I guess the formula that we need to know is what? First, pH plus pOH has to be equals to what? 14, right? Very good. So then the other one is pH or pOH equal to negative log of the concentration of proton. Okay, so with this, so we can start looking at the information given. So they say the hydroxide concentration is 0.00001. So in here we have concentration of hydroxide, right, equal to 0.00001, which is equals to 10 to the negative 5, right? So if we take the log, negative log of that, so we get pOH is equal to, okay, so we get that, so our pOH is equal to 5, okay? So if we get, now we can get a pOH of 5. Can we calculate pH now? Yes, right? Using this formula, so we have pH equal to 14 minus 5, so we get pH equal to 9. Okay, so the answer is E here. Okay? So next one. Okay, so the pool, the water in the pool had a pOH of 10. After adding a buffer, a pH is now 8. Which of the following is true? So we look at the information given. Again, we're starting off with the pOH of pool, right, equals to 10, okay? But it's going to be very difficult to compare pOH to pH. So we need to convert them into the same scale. So now the pH of the pool would be equals to 14 minus 10, which is 4. So now we know that initially the pool has the pH of 10, but after the buffer, we have the pH of 8, right? So after we put the buffer in, we have pH of 8. So is it getting, the pH is getting higher, but the thing is, are we is it becoming more basic or acidic? Basic, right? So now if you look at the choice here, initially water was basic. Is it correct? pH 4, is it basic? No, so that's out, right? So the next one, the water become more acidic. So from 4 to 8, is it become more acidic? No, so that is not correct. The pH of the water decrease? No, it's increasing, right, from 4 to 8. So that's not right. So the concentration of hydrogen increase or decrease. It's the last two choices. So if it's hard to look at this, so we can actually convert it back. So now with this, we're going to have the hydrogen, the concentration of this equal to 10 to the negative 4, right? For this one, we'll have negative 8, okay? So is it increasing or decreasing? Which one is smaller? Negative 4 or negative 8? Which one is smaller? Negative 8. So negative 8 is smaller, right? Hmm? So if you look at this one, 0.0001, that negative 4, right? 0.000, I don't know how many of them, 6, 7, 8. Which one is smaller? 8, negative 8. So this one is smaller, so it's decreasing, right? So the answer is E here, okay? Right. All right. Okay. I don't know if it's better or worse. Okay. So use the following information to answer question 6. Paul is a laboratory technician and is assigned to do some tests on a patient blood sample. He's supposed to drop red blood cell in 70% solvent, with 70% solvent into a beaker with the solution of 30% solute. Okay. However, he didn't have enough time preparing for the test, so he stood up and he accidentally dropped the sample in a solution with 30% solvent instead. So the question 6 asks, after the accident, the cell would be blank compared to the solution in a beaker and blank under the microscope. Okay, so what we're going to do here, so with this, usually we need to draw, right? So we need to draw. You see, we have the beaker and we have the cell. Let me see. Let me put this in this different. Okay. So with the information given, what is the percentage of the solvent in the red blood cell? We have 70, right? %70 solvent. Solvent is what? water, right? H2O. So, and he said the accident here, after he accident dropped into the solution with 30% solvent, okay? Solution is 30% solvent. So from here, we know that how to calculate how the water molecule move, right? So we have, if you don't remember, mole H2O is what? Mole H2O is hypotonic, right? So which one is hypotonic? The cell, right? Because it has more water. So they move the molecule, and it moves from high concentration to low concentration, right? So we have from cell, the water within the cell move out. So the first one is the cell would be hypotonic. So A would not be correct, right? C would not be correct, and E is not. We can just cross them out, okay? You should do the same thing when you do the test. Kind of helps you, you know, like eliminate the, you know, the wrong choice. And then if the water is going out, the cell is shrivel or is it burst? Shrivel. So the answer is B here, okay? So you need to know the different term, shrivel, shrink, you know, the synonym of those, you know, could be used on the exam as well, okay? So this one is more of the definite memorization, but I just throw it out. She might ask you something about this. What is organic chemistry? Study of organic produce? Probably not, right? So this one, I'm just gonna answer real quick here because it's all memorization. You need to know that. The study of carbon compound, okay? Next one. Which of the following is not a part of amino acid structure? So amino acid structure, we have carbon, right? And we have one H, we have COH, we have, you know, NH2, and we have R. So that's pretty much the amino acids. So we have C, we have H, amino group, this one. We have carboxyl, so everything is part of this amino acid, so the answer is none of the above because all of them are. Okay? So next, which of the following is true about the protein structure? Okay, so first of all, I can go through this a little bit about the protein structure, kind of review. So we have how many levels? Usually we have 3 levels, but some protein might have 4, right? So we have primary, secondary, tertiary, and quaternary. Okay, so let me put here, see, protein structure. We have primary. So primary would be, we would have amino acid, right? And bind as a string. So what happened between amino acid group is we have the peptide bond. So the key here, in this one we have peptide, right? So what else that happened? So when during the, they make the peptide bond, so we have we use the dehydration, right? So dehydration also known as, anybody? Condensation. Okay. So then after that, we're going to have secondary. In secondary, what's happening? The key things here is, what's that? Folding. Very good. So the key here is start to fold, right? It's folding. So what kind of bond that help us with this? Hydrogen bond. Okay. So hydrogen bond, and what else that happened here? There's two types of how the structure, the secondary is formed. We have alpha helix, right? And beta sheet, right? So we have those two. Then after that, we have tertiary, okay? In tertiary, what's the major things here? Interaction between our groups, okay? So when you have this amino acid, each of these is this one, right? This is your amino acid, right? So each of these, sometimes you might have R group hanging out. So, and the interaction between R groups is what's happening in tertiary, okay? So we have the, I'm just going to put interaction. R groups, okay. And the last one is for quaternary. It's just when you have two or more polypeptide. Okay. So all of this information should help us answer this question. I mean, I'm just throwing this out, trying to get you the most, information as possible. So which of the following is true about protein structure? So when denatured, it loses the primary, secondary, and tertiary. Is that true? No, because right here, the primary stay the same. We lose only secondary and tertiary. Okay, so that's not right. So hydrogen bonding is involved in level 2. Is that correct? Right here, right? So that one is correct. So if A is incorrect, we also cross that one out. So for C, the interaction between R group occurs in tertiary level. That's also correct, right? So we have this. So the answer is B and C. Okay. So right here, when I have wiped out here, so it's basically I had some typo on there. So which of the following protein structure level would be affected by chaperone? Anybody remember what chaperone is? Chaperone is a protein, or the protein that help in what? Folding. So if it's folding, we have the key word here, folding, right, earlier. So folding is what level? Secondary or level 2? So the answer here is level 2. Okay? So now, so 11, we have gap junction are most likely found in what? So if we look at three different type of cell junction, right? We have tight junction, we have gap junction, and the anchoring junction or desmosome. So the first one, tight, if you recall, the tight junction make the cell like really tight together, so water doesn't leak. So that would be something that you found like in your stomach, right? So what happened if your stomach leak? So the acid will come out and it's going to harm us. So the first one definitely not what it is. Okay. So the next one, the skin, the heart muscle. Why heart muscle? Is it gap junction? What does gap junction do? I'm sorry? Transport it rapidly. So it basically has the channel that when you talk about the transport, the channel that can communicate between cells, right? So what would happen if they don't, if your heart doesn't, your heart muscle don't communicate well? They don't. The pulse doesn't, synchronize, right? So muscle from the left, maybe on the left or the right, are not, going well together. So this one is actually the answer here. The heart muscle is where you found gap junction. Skin would be desmosome. Okay, so that's the answer for that one is B. Okay, which of the following is most likely affected on the cell membrane fluidity? Anybody? Cholesterol. Okay, so this one is sort of memorization, but at the same time, to explain a little bit, if you have the, this is the bilayer, the phosphates, right? So this is how your the membrane look like, right? So if it's if it's low temperature, so all those would be really tight. So if it's too tight, so it's going to be forming, like make your skin very tight and dry. So that is make it very rigid. It's not fluid. So what it does is the cholesterol will go in between each of this. Well, maybe it's not like it doesn't have to be in every all of this. So it can be random, and then kind of split this apart a little bit, so make it less rigid. Okay, so it's more fluid. But at the same time, when the temperature is high, so the gap between this can be really wide. So this cholesterol would fill in some of those gaps and make the two molecules closer together. So that also deal with the fluidity of the membrane as well. Okay. So for 13, blank goes through cell membrane most easily. So definitely the first one, small, right? It has to be small. As we know that we have a two-part, hydrophobic and hydrophilic. So if it only one of those, it's not going to go through easily. So actually this one you have to have what? You have, it doesn't, if it's hydrophilic, can it go through? Because which one is hydrophobic part? This one is hydrophilic, right? Because it has polar. But this part is hydrophobic. Okay, so even if we get through the first part, the hydrophilic part, but once it's get into hydrophilic, it's gonna get repelled. So it can't really get through. So this one, only what we get through is these two. So the answer would be the A and C here. So hydrophobic and small. So next one, also part of it, you know, I guess the memorization. So you probably don't have to go into detail of the, you know, of this, but you need to know which one is the first, which one is the second law in thermodynamic. Again, this is another area of typo. Just we know that the first law of thermodynamic is that the universe is moving toward greater entropy. That one is actually the second law. The first law is what? Energy cannot be created or destroyed. So this one, the answer would be false because this is not the first one. Okay. For 15, which of the following is true about exergonic reaction? Okay, so what do we know about exergonic? Exergonic is basically release energy, right? So exergonic is releasing energy, so delta G is negative. It's also, we have, if we have the delta G negatives, so we know that this is going to be catabolic. Okay. What else do we know? We have because catabolic is what? It's building or making molecule or making bond or making molecule. So we have hydrolysis as well. Okay. So the thing is, I kind of pointed out all of this because if you know exergonic, you can also know endogonic. You just have to know one thing. The other is the opposite. Oh, sorry. Yeah. I apologize. You're right. Breaking. Okay. Thank you for catching that. Sometime, well, you know, sometime I also make mistake. Okay. So with this, so we have so many of them, so, but we know that for sure the first one is correct, the second one is not, right? So if delta G is negative, is it spontaneous? Yes, right? So this one is also correct. So the answer is A and C. Oh, sorry. Again, I apologize. So it's spontaneous, so the non-spontaneous is not the answer. So the answer is A. Okay. I get nervous, you guys. Look how many people are here, okay? I get nervous. Okay, 16, which of the following is not true about glycolysis? Oh, that's a whole thing. So let's look at the information we have. The first one is it substrate level phosphorylation. Is it true? This is true. Enzymes are required. So if it's substrate level, it's also required enzyme, right? So does it occur in matrix? Glycolysis? So this is the problem. It's not in metric. Where is it happening? Cytoplasm. Or cytosol, right? So the answer is this one. So let's look a little more. The product includes 2 pyruvates. Is that correct? Yes. Right. Okay. So 17, right, catalyzes the transfer of a phosphate group from ATP to another molecule. So this one is actually one of the things that you have to memorize. So is it MAD? What does MAD do? MAD is the gene responsible for the hooking up chromosome with spindle fiber. Okay, so this one has nothing to do with the ATP. Kinetochore. What is kinetochore? Okay, protein on centromere. Okay, again, this one is also related to, how the spindle fiber attached to the centromere. So this is not correct. RNA polymerase is not related. Ligase is not related to this. So the answer is kinase. Okay. So again, I ran out. I just keep using the easy one that comes to mind. Operate a P with heterozygous genes for large size, green color, and smooth skin, with another P of the same gene for size and color, but not the skin. So due to his clumsiness, he dropped his laptop and he had to do, run report manually. So we need to find out how many Punnett square boxes does he need for this experiment to find out. Okay, so what rule do we use here? We use 2 to the n rule, right? So the first one we have large size, green color, and smooth skin. And they said this one is all heterozygous. Okay, so if we use large size as L, heterozygous, right? So in green, and the skin, let's say S. Okay, so this is the first P. The second P, we cross it with, they said the first two, they said the same gene for size and color. So size and color is the same, but not for the skin. Okay, so the rest are heterozygous. So the skin would be homozygous, but we don't know whether it's, like dominant or recessive. Does it matter here? It doesn't, because it's going to be the same. So because 2n rule, n is from what? The number of heterozygous. Very good. So we have how many? We have 3, right? 2, 3, and 2. This one, let's say, we can just put small. Okay? So we have 2 to the 2. So 2 to the 3 is 8 times 4. We have 32. Okay. Okay, 19. If 2n equal to 20, what is the ratio of the number of chromosomes in anaphase to the number of chromatid after cytokinesis? 1. So the key part here, you have to be able to identify when they say anaphase 1, is that in meiosis or mitosis? It doesn't have any number, so it's automatically a mitosis, right? Because only meiosis you have 1 and 2, right? So this one we talk about mitosis. Okay, so if we talk about mitosis, so mitosis, if we start in, let's say P, It's prophase, metaphase, anaphase, telophase, or cytokinesis. Okay, so we start with 20 chromosomes. 2n is equal to 20. So how many chromatids do we have? So anyway, so we don't really need to know about that because we're looking for chromosome, right? So we have 20 for prophase, we have 20 for metaphase. What happened in anaphase? Split. We have it split, and each individual chromatid become chromosome, right? So at that point we have 40 chromosomes, okay? So now we know how many chromosomes we have here, 40. Okay, so now next we have MEI, meiosis. Okay, so meiosis we have prophase 1, metaphase 1, anaphase 1, and 1. So for meiosis, we start with 20, but now they ask for what? The chromatids, right? So the chromatid, so we have to think about the chromatids here. So now the first one we have 20 chromosomes. How many chromatids? We have 40, right? Because they are replicated, okay? So same thing. Anything happen? What happened in the anaphase one? Anything happen? Okay, nothing. So it's what split in anaphase 1 was the pair, not the chromosome, right? So it's still like that, the same thing. So, but after that, remember meiosis. The first one, meiosis 1, the purpose is to what? To make haploid. Right? So if to make haploid, so this one has to be half of what we have, right? So that one is 10 chromosomes and 20 chromatids. This one is 10 chromosomes with replicated, okay? So the answer to this, we ask for chromatids, so we have 20. So the ratio to this is 40 to 20, which is 2 to 1. Okay. So next, which of the following is not true about meiosis? Okay. Synapses occur in prophase 1, is that correct? Yes. Right? Each chromosome line up at the metaphase plate in metaphase 1. Is that correct? This one actually is not. What happened in metaphase 1? Is that the chromosome line up or the pair line up? Right? So be careful. So this is not each chromosome, it's the pair. So that one is not correct. So now we know the B is not correct. A is correct, right? So if A is correct, so this, all this are gone. Right? So for C, after meiosis 1, daughter cell go through G1 only before entering prophase 2. Is that correct? Yes. G1 only, not G2. Okay. So if C is correct, so that one is not. So we have to look at the last one. For each anaphase, chromosome are unreplicated. Is that correct? So don't forget, with this, is there unreplicated for anaphase here? So right here, it's anaphase, but still is replicated. So this one not always true. So the answer is this one. Not always. What's that? Oops, sorry. Okay. So that one, not always. Next one. Which of the following does not occur in mitosis? Okay. So is centromere separated? Yes. Cleavage furrow can be found in telophase. Correct. Right. Spindle fibers start forming in prophase. Yes. Nuclear envelope disintegrate in telophase. Is it disintegrate or start forming back? Remember, telophase is pretty much the opposite of prophase, right? So in prophase, the nuclear envelope starts to disintegrate. So in telophase, it has to be forming back. So this one is not correct. Okay. Each chromosome is unrepresented. Okay. All right. So next one, it's like running. It's 50 minutes already. So I wonder, you guys want to take a break? Oh no. Okay. I'm just gonna keep going. Okay. All right. So if 2n is equal to 8, there are blank chromosome and blank chromatids. Okay. So if 2n equal, so we have, this is mitosis, right? So we have prophase, metaphase, anaphase, and so we start with 2n equal to 8. We have 8 chromosomes, 16 chromatids, right? So then anything happen? Nothing, right? So then in anaphase, this one, the chromosome split, so we have 16 chromosome and 16 chromatids. And then after that we have this. But they only ask this in anaphase, so we have 16. Okay. Next one. Karyotype can tell if a person has a blank. So anybody remember what karyotype is? A picture of all the chromosomes. Very good. So that one would tell us many things. You know, can it tell us if the person has Klinefelter syndrome? Yes. Because what is Klinefelter syndrome? It's a male that born with more than one X. Okay. So you can have XXY or even more. You can have XXXY. That's Klinefelter. Okay. So since it shows all the chromosomes in that picture, so if it appears that you have more than one X and one Y, so then you can tell if the person has a Klinefelter. What about Turner syndrome? Turner syndrome is for a woman with only one X. Okay, so you only have one X. Okay. Down syndrome, you have triple of the chromosome number. They call it trisomy of chromosome 21. Okay, so all of this you can see in there. So the answer to this is A, B, and C. You can see from the karyotypes. Okay. Okay. Next we talk about, we have this, small and green homozygous recessive, and heterozygous big and red bean is crossed with another heterozygous big and red bean. So if we put down here as SS, refer to small, and G is to green, right? So we have this, and then they say heterozygous cross with another heterozygous. So we're going to have cross with this. Okay? So what happened here is what? We have, again, we can use 2n rules, right? 2, 2, 2, 2. So we have 4 times 4. We got 16. So, but the thing is, this one, if you know, they have what? The rules of 3, 3, 3, 1, if you recall, right? So this would be homozygous, you know, the dominant one, and this one would be the heterozygous. This one, the last one, would be the homozygous recessive. So if you look at this, a question as, what phenotype will you most likely found in the offspring? Okay, so this one is the one that is most likely found, right? So the most likely found here would be this one. Like I said, it's going to be heterozygous. It's going to be big, right? It's going to be big, and it's going to be red. So the answer to this is big and red. Okay. So this one, if we look at this one is basically big and red would be 9. Big and green would be 3. Small and red would be 3. And small and green, which is the recessive here, is the least likely found. Okay. So this one, if you remember before you go in to take the test, it saves you a lot of time instead of drawing the 16 boxes, right? So it's very important that you know this. This only happens when heterozygous, when it's that's the only time that we can use this ratio. Okay, what is the probability that the offspring will be small and green? So once we have this, we already know small and green from here. So the answer is 1/16. Okay. The next one is also, you know, the simple one. So once we have that ratio, we can answer this many questions. So the probability that the offspring will be is 3/16. So how many of them we have 3/16? We have this one, right? We have this one and this one. So small red and big green. Small red and big green. So the answer to this is B and C. Okay. We have this. Now I guess this is your favorite question. A munchkin cat, right, is an autosomal dominant trait in cat that is lethal in homozygous dominant condition in embryos. So if two munchkin cat mate, what is the probability of the genotypically normal kitten? So munchkin cat is this, right? So mate with this. So what we have here, we can have a box like that. So one here, right? So crosses, we have big M, big M, big M, little m, big M, little m, little m, little m. Okay? So what happened here is it said it is lethal in homozygous dominant. So this one is out because it's not born. Okay? So they ask the probability of genotypically normal. So which one is genotypically normal? This one, right? Small, small. So then the probability is 1/3. Okay? So this one, again, right here, I put here, that's a typo that I have. So two poor cats are in mating season. A male calico cat with Klinefelter syndrome, but with a Turner syndrome orange female cat. So what is the probability, or what is the chance of having orange male offspring? Okay, so we know that the Turner syndrome has only one X, right? So you can only have one X on here, and it said orange, okay? So orange female, so we have that one, XO, orange, okay? So we have Klinefelter syndrome, you have more than one X. So instead of have just a regular, just one X, we have two X's, and we have Y, right? Because Klinefelter happened in the male. So, and they said the Klinefelter is calico, so it has orange and black, right? So what we have, we're going to have XO, 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 XB, and XOY, right? So the chance of having an orange male offspring. So this one is orange and it's male. So the answer to this is 100%. Okay. So it is not 1/3. Why? Because we already know for sure that, but because it talk about male, every male will be orange, right? So that means it's 100%, not 1/3. Okay. Yes. Which one? This one? They'll only ask for male. Okay. All right. We get it squared away. Okay. So next, colorblind is homozygous recessive and expressed on X chromosome. So what does it mean it's expressed on X chromosome? That means it's sex-linked, right? So when the question related to sex link, so you have to use X and Y and the superscript, right? So if we use this, we have homozygous recessive expressed on X chromosome. A colorblind man marry a woman who is a carrier for color blindness. So a guy, right? And the woman, okay, the guy is homozygous, has a guy is colorblind, so basically just one is enough, right? Because it's expressed on X. So the woman is a carrier, so only one of the X has B, so they cross. So what would happen is we have this, right? So we have XB and X and XB and Y. So we have XB, 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 X, XBY, and XY. Okay, so it asks, what is the probability that the newborn will be carrier? Which one is a carrier? This one. Is this one a carrier? No, right? Because it's the guy, so you only have one X, then you have that. So it's not. So the answer to this is 25%. Okay? So which of the next one, which of the following play an important role in male sex determination in human? Okay, so this one again, it's memorization. I just throw it out that you need to know what it is. So FYI, for your information, of course it's not. AGG, MAD, we talked about it, ATP, adenosine triphosphate, your power. So this one, the answer is SY, okay? Don't you just love this kind of problem? A, C, A, and B, that kind of thing. So, which of the following is true about a cat with Turner syndrome? Okay. So, Turner syndrome, it can only be female. Is that correct? Yes, because you can only have you have only one X, right? So it's only be female. Can it be male? No. Can it be calico cat? No, because to have calico cat, you have to have what? Orange and black, right? So since you only have one X, you get to choose either black or orange. You cannot have both. So this one is also not correct. Okay. So let's say X, or let's see. It can be a carrier for homozygous recessive. It cannot be, I'm sorry. It cannot be, is that correct? It cannot be, that is correct, because if you have only one X, if it appears, you have it, right? So it cannot be a carrier. So it will have the disease, basically. Okay, so the answer, so since B and C are incorrect, B and C, B and C, so the answer is this one, A and D. Okay. For 32, if guanine is 15%, what percent is thymine? So that's pretty simple. So we have guanine of 15%, right? So guanine complement with cytosine. Cytosine is also 15%, right? You remember that we have AT and CG, right? So this one, each of this has to be equal. So since this two is already 30%, we know that altogether we have to have 100%. So the leftover for A and T would be 70%. So 70% for A and T. Therefore, each of this is 35%. So the answer to this for thymine is 35. Okay? So which of the next one, which of the following cannot be a DNA? What's the difference between DNA and RNA? DNA we have A, T, C, G, right? For RNA we have A, U, C, G. So in any of this, if it has U, that is not a DNA. So this one has T, DNA, 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 DNA. So this one is not DNA. Thank you. Okay. Next one. Which complementary is associated with 3 hydrogen bonds? So this one is, you need to memorize, memorize that. So all of the AT, AU, this one, is this possible? CT? No, this one is not possible, not possible. So we have this. So this one is 2, so this one is 3 hydrogen bond. So the answer is E. Okay. A nucleotide consists of the following except, so we have nitrogenous base, we have phosphate group, that's correct. Pentose, which is the same as 5 carbon sugar. Okay, so these are all, yes. Which one? A, C and D? I'm sorry, I can't hear well. Oh, because they're not complement to each other. C and T. Which? Uh-huh. Yeah, because the question asks which one is associated with 3 hydrogen bonds. Oh, why? Oh, because, well, I guess because the way the molecule arranged, you know, they have 2 rings. I have to go into, I don't really have the book, but if you go back into, like, and look at the slide, so between C and G, because of how it arranged, it will form the hydrogen bond for 3 of them. Okay, especially I think it's good that you asked, because I think actually she mentioned a lot of this. You know, many times that the book had it wrong when they have the dotted line to represent a hydrogen bond. So I don't know if she's gonna ask it again. So just to, you know, it's more like, it's very much of the chemistry stuff. I don't think she's gonna go into detail, ask you why, because it's not much of the biology, okay? But you just need to know that C and G has 3 hydrogen bonds, AT or AU has 2, okay? All right. Okay, did I make myself clear on that? Okay, so we have next one we talk about that all of those A, B, C are the you know, the component of nucleotides. So this one, I guess, is they said except, right? None of this. So this is because all of them are. So the answer is none of the above. So this one, this use information, okay, I borrow the name from somebody else. Fakwa, a biomedical science student going to medical school full-time and work part-time, stay up late, did not have enough sleep, trying to finish his assignment. During genetic lab, he needs to replicate DNA from a sample, and there are several enzymes involved. Due to sleep deprivation, the first 2 attempts were not successful. So the question asks, what did he forget to put in the first attempt if the DNA is fragmented? Okay, so instead, since I list all of these possible enzymes, so we're going to go through what each of these does. Okay, so helicase, what does it do? So it's basically split the DNA, right? The DNA strand has a double strand, so helicase split it by breaking the hydrogen bond that we've been talking about, 2 bonds or 3 bonds for each of those are complementary. So the split DNA strands, right? Next, single-stranded binding protein. So it's basically what it does is to pin down the single strand DNA, okay? So one is split, so it becomes single strand, and this single stranded binding protein just pin it down, so it won't go back up and, you know, create the hydrogen bond again. So next one, Topoisomerase. Okay, what does it do? Anybody? So this one is the one that she usually asks, because it's kind of the unique in a way that this one does the prevent spinning at replication 4. Okay. So prevent spinning or coiling back at the replication 4. Sometimes we might use this relieve the stress or the strain at the replication 4. Okay. Primase. What does primase do? Put down primer. Okay. So can we do synthesis without RNA primer? No, right? So that's very important. So DNA, next one, DNA polymerase 1, what does it do? What's that? Can you say? Yeah, so very good. So basically, it's remove, right, RNA primer and replace with DNA. Okay, I just put it in different order from what it's supposed to be, but I guess it kind of helps us to make sure that we know exactly what each of these enzymes does. What about polymerase 2? We have this one does the proofread, right? And repair if it needs to. Okay. The polymerase 3 actually is the one that does the DNA, synthesis, or let's say to put down the DNA or DNA synthesis, I guess. Once again, how do we make DNA? Which direction? 5' to 3'. Okay. Very important. Okay. That also true for RNA as well. Okay. The last one, ligase. What does it do? Seal the gap of Okazaki fragment. Okay, so we all know all of this, but this one is not in order. Make sure you know the exact order that it goes, because she might ask you, switch it. She might switch it around and ask you to arrange the order of, enzyme, that kind of thing. So you said if the question asks if the DNA is fragmented, that means we didn't have anything to seal the gap, right, in here. So that what did he forget to put in the first attempt? So that that's why DNA is fragmented. So the answer here is ligase. Make sense, right? Okay. Okay. So next one. Yes. Explain what? Topoisomerase. Okay, let me do this number 37 at the too, and you will see why like how it does. I'm just going to draw here to help a little bit. Okay, so let's see number 37. This guy again. Forgot to put one enzyme in the second attempt, but the label was not legible. He's certain that the helicase, single-stranded, and topoisomerase were added. What is that enzyme if the long single-strand DNA has no complementary at all? Okay, so if we look at this, right, this is how, this is after the, the DNA split, right? So as a result of the helicase, then the single-stranded pin down each of these single strands, okay? And here is where the C, which is topoisomerase, comes in, okay? Here is where topoisomerase, this is where we call the replication fork, okay? So just like you're trying to split them away at the strain at that fork, and then it's trying to spin back. So that is the job of topoisomerase, trying to relieve that strain, okay? Okay, so then what happened here is after we have that, and then they said there's no complementary occur in each of these strands at all. So which one did we forget? So ligase definitely not, because we don't even have DNA in it, right? 2, 3, this one we don't have. So 1, you don't even, you can't even remove it. So the answer here is primates. Because to be able to make any DNA, you have to have primer first, right? And then you make more of it, okay? But now you have nothing. Your single strand is like nothing, complement, no complement. So the primase is the answer, okay? So next one, which of the following is not true about DNA synthesis? Okay, so we talk about this. If you look at this, if it's continuous, let's say this is, I guess this is going to be 5'3". This. So this one, if you look at this, the synthesis is like this. Okay, this one. Is this one single, is this one leading or lacking strand? Leading, right? So how do you know it's leading? Because the synthesis is continuous, right? For lacking, what happened? Lacking, we have, it's fragmented. Okay. So what else? What happened? The synthesis is going from which direction? This direction, right? No disruption. So this is outside in. For lacking is inside out. So if you look at this, the answer to this synthesis for is continuous on lacking strand. So that one is not true. Okay, so I guess we have to circle that one. Not true. And the leading strand is inside out. Okay, that is also not true. So the answer to this is B and C, right? '39. What enzyme prevents spinning at the replication fork? We already covered that, and we just talked about that a while ago here. This one. Topoisomerase. So this one I don't even actually have to go even more, right? So topoisomerase. DNA synthesis occur in direction of 3 to 5. Is that correct? So it's is from 5 to 3, right? So actually this one is false. Okay, next. Which of the following is not true regarding transcription? Okay, so the first one, RNA polymerase produces pre-mRNA. Is that correct? Yep. Right. So it produces pre-mRNA, not mRNA. Okay. So this one is true. Tata box is located in the promoter region. Right. RNA polymerase bind at Tata box. It's also correct. Transcription occur in cytoplasm? No. This one, where does it occur? Nucleus. Very good. Okay. The goal of transcription is to make mRNA. That's correct. Why is that? Because where do we find DNA? Nucleus, right? It's not usual if we find DNA outside of nucleus. So that's the reason why we have mRNA, because M stands for messenger, right? So since DNA cannot be outside of the nucleus, so we send the messenger out. That's why we send the mRNA out instead of, having DNA out. Okay, so that one, the answer not true. The answer is this one. Okay. So 42, which of the following is not true about transcription again? So pre-mRNA includes introns and exons. Is that correct? Right, so this one is also correct. Splicing of exons is required to make an mRNA. Is that correct? Actually, it is not. What's wrong with this? Splicing introns, right, instead of exon. So what's the difference between intron and exon? Exon what? Exon include the coding, right, the codon. Intron don't. Okay, so that's why we need to cut it out. The process of cutting, we call it splicing. Okay, so this one is not true. So just go through a little bit more. 5-cap is needed to make a complete mRNA. That's correct. Poly-A tail protects mRNA in cytoplasm. That's correct. Okay, and introns contain regulatory genes. Yes, it's a regulator. Also, if you recall, like what we talked about earlier, this promoter region. Does promoter region contain any codon? We cut it, so the promoter region is also a control region, just like introns. So if she asks something, is promoter region the same as intron? The answer is yes. Okay. The following statements of the translation are true, except, so if we look, the first one, ribosome consists of small and large subunit, correct, right? The large part has E, P, and A site. That's also correct. Okay. mRNA dictate the amino acid group. Is that correct? If you're not sure, we can leave it. Okay. So tRNA has anticodon. So this one, tRNA has anticodon, is correct. mRNA dictate amino group? Anybody? So actually this one, I guess it, this one, it's also actually the dictate the the group. So this small, let's talk a little bit about large and small. Since a large subunit has EPA, so the mRNA actually is the one that dictates, but tRNA has anticodon to match, so to make sure that it's correct. So I might have this one. I might have all of this is actually are true. So there's all of these are true. So nothing is really incorrect here. Okay, so now I don't even. They said except all of them are true. How do we answer this? None of the above, right? Because all of them are true. So I'm going to add. Okay. Now we have an answer. Okay. So next one, which of the following is not true regarding translation? tRNA matches its anticodon with codon and release amino acid. It's correct, right? So next one, all tRNA binds at AP then E. Is that correct? No. The problem is the all part, not all. Only what? Only the second, only the second and on. The first one, tRNA bind to what? P and E only. Okay? Because if it binds to A, then the next, we don't have space for the second one to come in. So it binds to P first. Okay. So this one is not true B. So multiple ribosomes can process on one mRNA at the same time. Is that correct? Anybody? So this one is also correct, actually. So the answer to this is B and C. Okay. So I think I might have this one wrong, these two wrong on the answer key that I posted on the WordPress website. So the answer, that one is, this one is, B and C. Oh, again now, I'm sorry. What did it say? Not true. That one is true, right? So I apologize for the gazillion times. For this one, the answer is B. Because A and C is true. You guys making me nervous. Over and over. 44, 6 more questions. Okay. All right. Next, which of the following is not true? Again, two identical sister chromatids are attached at centromere. Is that true? Yes. Centromeres are on metaphase plate during metaphase. This is true. Because you have to split it, right? So it's in the middle. It has to be right there. Centromere can be separated in cell division. Of course. Otherwise, you cannot separate the chromatids. So kinetochore is a protein that sits on the side of centriole. This one is a problem. This is centromere. It sits on a centromere, not centriole. So this one is not true. So the answer is D. Okay. So next, cells in nerve tissue stay at G0. Is that correct? Yep. Stop. It stop dividing. It doesn't divide. So the answer is true. Missense mutation produces a premature stop codon. Is that correct? Yes or no? False. Why is it false? It's nonsense. Nonsense mutation, I didn't say it's nonsense. Okay. So this one is nonsense. Okay. 48. Which of the following is not true about TCA? Okay, the first one. What is TCA again? Anybody remember? TCA is Krebs cycle or citric acid cycle. Okay, so you need to know all of those. So first, pyruvate are brought into mitochondrial matrix by Tim and Tom facilitated ports. Is that correct? All of this correct except, again, one part. What is Tim and Tom? Active port. So facilitated is not active, so that's not correct. Okay, so every little details, we have to go through. I myself make mistakes several times. Citric acid was formed by a pyruvate and oxaloacetate. Is that correct? This is correct until here. Citric acid was formed by what? Acetyl-CoA and oxaloacetate. So that one is also not correct. For each cycle, NADH, ATP, and FAD are obtained respectively. Yes, we have all those three, right? So the answer to this is, so you talk about not true, so this one is A and B. They're not true. Okay. So next one, this one, I guess this is the last two questions related to this, and we're going to need to use this. Okay, so the question 49, determine a series of amino acid from DNA below. Okay, so with this, again, what each of this is? Codon. Where is codon? Where can we find codon? Is it in DNA, tRNA, or mRNA? mRNA. So this one they gave us as a DNA. So we have to convert this into mRNA first, right? So mRNA with this, this one kind of tricky. If you look at this, you don't see any T. You might have thought that this could have been mRNA, but it's not if we didn't read this part, right? So make sure, there might be something like this on the exam and you thought, oh, there's no T, you just jump right into the table. That could be a disaster. Okay, so we look at this. So if we look at this again, it's gonna we're going to have the anti-parallel, right? So we're going to have what? CCG. What we're going to get is G, G, C, right? And next is C, C. Is that T or U? U. And we have U, C, C, and C, U, C. Okay, so with this, we just, after that, we can go and look for each of this in this table. Okay, let me kind of zoom in a little bit, if we can. Okay, so with that, the first one, GGC, where's G, GC, right here, right? So the first one is gly, so it would be either this one or this one, right? So the rest you can actually just cross it out because the first one is not consistent with what we found. So the rest are, to save some time, the rest are pretty much the same except the third one. So the third one is UCC. Where's UCC? UCC. We have this, sir. So we have this one. So this one is not correct. So the answer is B. Okay. Question? Okay, last one here. So determine the serial amino acid from the mRNA. Now, so we have mRNA. Do we have to convert it? No. So this one, like I said, make sure you really pay attention because it could be, interpreted incorrectly. So we have UCA, start with UCA. UCA is this one, start with the SER, right? This one, UCA. So we have UCA, start with the SER. So then the first three are out. Okay? So what's the difference with this one now? This one, the third one that are different. So we can look at AAA. Where's AAA? AAA here is LYS. So the answer to this one is E, LYS. Okay. I guess that's pretty much what we have. But I mean, if you have questions for the last 15 minutes, I'm more than happy to, help you with anything for the last 15 minutes. Okay, and then best of luck on the exam tomorrow and subsequent days. And hope you have a great break.
Thank you for that.
Thank you.
Thank you. Mm-hmm. Have a great break. And
you too. Thank you. I hope that helps. Okay. Thank you.